This is Chemistry of Carbons and its Compounds, Part 5. At the beginning of this presentation, we'll look at a couple more addition polymers. Then we'll look at condensation polymers. And then we'll talk about the recycling and the need to be environmentally aware about polymer production. We'll also talk about the formation of pharmaceuticals. The assessment criteria is as follows. Know a range of properties of polymers and understand how these are related to its structure. Know the linkages and repeating units for polyamides and polyesters. Know a range of condensation polymers and some of their common uses. Know the importance of synthetic polymers in modern life and know a range of common products which are made from them. Understand the environmental disposal difference between addition and condensation polymers, and appreciate the advantages of recycling these polymers. Appreciate the range of compounds which have physiological actions. Understand that many pharmaceuticals occur in nature but can be synthesized. Know some examples of synthetic drugs. Understand the importance of molecular shape and chirality in physiological action, and understand the problems of drug delivery of pharmaceuticals and some simple solutions. Know a range of properties of polymers and understand how these are related to its structure. In the last presentation, we looked at some addition polymers. We really didn't talk about what were done with them other than how to create them. But there are things you can do with these polymers to make them behave or make them into the things that you need for use in our modern society. For example, polystyrene beads with a 5% volatile hydrocarbon attached in a liquid form causes the expansion of the hydrocarbon when you heat it and the subsequent expansion of the polystyrene to fill a mold being a shape that you want to make the polymer into. That's called expansion molding. There's another kind of way of making polymers into shapes that can be used. Another one is thermosetting. When certain plastics are first heated, bonds are created across adjacent chains of polymers. So one chain is connected to another chain. If you do that, thermosetting, the process is called, that prevents the plastic from melting when reheated. It tries and keeps its shape. There also are network polymers. When monomers are created into polymers, but they're done in a three-dimensional way, this cross-networking forms a very hard plastic. Costume jewelry, the pieces of plastic that are cut into pieces for necklaces and earrings, are examples of network polymers. Now there are condensation polymers. Condensation polymers are polymers formed by difunctional monomers to give a polymer and some small molecule such as water. Polyester is an example. Polyester is a condensation polymer that has two monomers or more connected through ester linkages. Kevlar, the things that police wear that stop bullets, is a condensation polymer that consists of a long chain of benzene rings interconnected by hydrogen bonds. Bakelite, which we talked about just in the previous slide, is formed from phenol and methanol. It again has a rigid structure. It's a condensation polymer, and as I said before, a thermosetting plastic. Know the linkages and repeated units for polyamides and polyesters. What I'd like you to do with this slide is to pause it in the presentation and look at it and then try to see the repeating patterns. I can describe those repeating patterns, but I think it is easier for you to look at the AB patterns for both polyamide nylon and ethylene terephthalate polyester. Remember, the repeating patterns are AB in both of the two polyamides and polyesters. Pause the video, take a look, and then continue with the video. Know the importance of synthetic polymers in modern life and know a range of common particles and products which are made from them. This is only a small number of the many kinds of synthetic polymers. I've talked about Kevlar, I've talked about Bakelite, but there are many, many more. We talked about last year Teflon, using a halogen as part of the polymer. But you can see there's neoprene. That's the kind of rubber polymer that is used for divers who go underwater. Nomex, Orlon, a kind of cloth, nylon, a kind of cloth, mylar, sheeting that is transparent, sometimes covers food. There's many, many common products. Again, stop the video, look at these, and get an idea of all the different things of which synthetic polymers contribute to our modern society. Understand the environmental disposal differences between addition, largely inert, and condensation polymers. Appreciate the differences and advantages of recycling these polymers. I've provided for you two flow charts that give you an example of how biodegradable plastics and non-biodegradable plastics can be recycled. Both of them can be chopped and shredded and washed and stored. But biodegradable plastics have other ways that they can be reduced and be made less harmful to the environment. There's a 10-minute video that a student did for her A-levels. And what we'd like to do now is to show you that video to give you a better understanding of those kinds of things and the information you need to know to be able to talk and be able to perform easily on the Cambridge examinations.
is a very large molecule made up from a repeating sequence of smaller units called monomers. For example, here is the monomer chloroethene. As you can see, it's unsaturated because it has a double bond. You need an initiator such as a peroxide, which has a weak oxygen-oxygen bond. This easily undergoes homolysis into two radicals under heat or light. A radical is a species with an unpaired electron. The first alkene reacts with this to form a carbon-centered radical. This adds to another molecule of the alkene, and so on. This is called addition polymerization, and incredibly long polymer molecules are formed by a chain reaction. So in the case of our example, the monomers add together to form polyvinyl chloride, or PVC. Paper has been tabbed as one of the more environmentally friendly alternatives to plastics. Yeah, it has its advantages. It's easy to print on, cheap to produce, and it can be moulded and coated, and it's light. But there is a raging debate going on as to whether, when you consider all the processes involved in the life cycle of both types of packaging, whether paper is actually the more sustainable of the two. One argument that you will often hear is that paper is recyclable, and so it must be better. Well, yes, it is recyclable, but so are many plastics. In fact, it takes 91% less energy to recycle a pound of plastic than a pound of paper, and it's not even bound to degrade quickly. This is down to pH and temperature. Also, according to a 2007 study by Bousted Consulting and Associates, it takes 4 times as much energy to produce a paper bag as it does a polyethylene bag, and 20 times as much fresh water. Most paper comes from tree pulp. Each year the USA consumes 10 billion paper grocery bags, requiring 14 million trees. Greenhouse gases are also produced in the manufacturing of paper packaging, as the process involves heating wood chips under high temperatures in chemical solution. So paper doesn't look like such a sustainable option now, does it? Many now argue that it's a myth that paper is more environmentally friendly than plastic. After all, both are recyclable, but both are a drain on natural resources. So what makes a polymer biodegradable? Green chemists have put together this set of rules to make a successful degradable polymer. Natural products are all biodegradable, so structures closely resembling natural materials are likely to biodegrade. This is why cellulose is used to make synthetic biodegradable polyesters. It is a polysaccharide, which is a long carbohydrate molecule consisting of a linear chain of over 10,000 glucose units. Catechol is an intermediate in the degradation of aromatics, so aromatic products that do not contain two adjacent unsubstituted or hydroxyl substituted carbon atoms will degrade more slowly. Highly electronegative groups, such as the nitro group, decrease the rate of degradation of substituted aromatics. Heterocyclic species are cyclic compounds that have atoms of at least two different elements as a member of its ring. These species are slow to biodegrade. This is because the degradation of these molecules involves a stepwise removal of the substituted groups, destruction of the rings, and progressive alteration of the constituent units. Highly branched aliphatic chains degrade more slowly than linear chains. This is because the alternative pathways required to cleave side chain methyl groups proceed more slowly. Aliphatic ether-containing molecules degrade more slowly. Materials containing strong carbon-chlorine or carbon-fluorine bonds generally don't degrade quickly. These bonds are shorter than other carbon-halogen bonds, and therefore they are stronger, and so lots of energy is required to break them. Materials that are water-soluble are likely to be biodegradable. Water-soluble polymers include certain polyvinyl alcohols or hydroxyethyl celluloses. Biodegradation rates normally decrease with increasing molecular weight because there is more material to break down. Another type of biodegradable polymer is the photodegradable polymer. These are designed to become weak and brittle when exposed to sunlight for prolonged periods of time. One way to bring this about is to blend the polymer with light-sensitive additives such as benzophenone, which catalyse the breakdown of the polymer in the presence of UV radiation. UV-absorbing groups such as carbonyl groups can be incorporated into the backbone of the polymer. These absorb light energy and break, fracturing the polymer chain and hence causing it to degrade. The polymers are initially converted to waxy compounds when exposed to light. They are then converted into carbon dioxide and water in the presence of bacteria. This last step in the process is biodegradation. Biodegradation is the chemical dissolution of materials by bacteria or other biological means. There are two main types of biodegradable plastics: hydrobiodegradable plastics, or HBP, and oxo-biodegradable plastics, or OBP. Both undergo chemical degradation first, resulting in physical degradation and a drastic reduction in molecular weight. These smaller, lighter fragments are then more susceptible to biodegradation. OBPs are made by adding a small portion of compounds of specific transition metals such as iron, manganese, cobalt or nickel into the normal production of polyolefins. These act as catalysts that speed up the normal oxidative degradation by factors of 10. HBPs tend to degrade and biodegrade more quickly than OBPs, but they have to be collected in an industrial composting unit. Both processes give the same end result: water, carbon dioxide and biomass. OBPs are generally less expensive, possess better physical properties and can be made with current plastics processing equipment. Also, HBP emits methane when it degrades under anaerobic conditions, whereas OBP does not.
Appreciate the range of compounds which have physiological actions. There are many carbon compounds that are used in the formation of medicines. You can see 14 in this list of functional groups that have physiological action. Anywhere from the simple alkenes and alcohols to tertiary amines and tertiary amides. There's a range of compounds that can be formed and that can be used by humans for good and for bad. Many pharmaceuticals exist in nature, but once they are extracted from nature, they can be examined and they can be synthesized, made in the laboratory. For example, the spice turmeric is an anti-inflammatory. We know that opium can be made to use heroin, an addictive drug, but in nature it is an analgesic. There are many drugs that can be derived from nature. Vitamin C comes from an orange, but we can also make it in the laboratory. So many of the pharmaceuticals could be taken directly from nature. They are, however, identified and synthesized so that we can preserve some of those natural products. The following video describes Vitamin Nature's.
I'm Barbara Timmerman. I'm a university distinguished professor and chair of the Department of Medicinal Chemistry. My research is looking for new drugs from nature. A lot of my research is done looking for new cures for cancer, for different diseases, infectious diseases, getting the chemicals from plants as well as microbes. The biodiversity is very rich, and there is a lot of organisms that have not been explored yet for new medicines. So my research group is involved in biodiversity prospecting, biodiversity exploration, looking for new molecules for the treatment of different diseases. There are plants in the world that are known for certain activities, such as ginger and turmeric. Those are plants that have been used for thousands of years as part of traditional systems of medicine. But we do not know much about their physiological effects, about their chemistry, so now is the time to study them. With the case of ginger and turmeric, we are concentrating our studies on rheumatoid arthritis and looking for anti-inflammatory compounds. Nature serves as a template for the discovery of new drugs. For example, aspirin was found in nature, and now aspirin can be synthesized very easily. It's a very simple molecule. But we have drugs such as Taxol that is a very complex chemical structure, and this molecule is important for the treatment of cancer. So we need to go to nature to get the original compounds.
Understand the importance of molecular shape and chirality, stereochemistry, in physiological action. About 50% of pharmaceuticals are stereoisomers. And of those 50% of the pharmaceuticals, another half of them have both N-antiomers, both left and right, present. Now, these two N-antiomers of a chiral drug may differ significantly in how they affect the body. So it's very important that in the synthesis of these drugs, that if there is a danger to human function, that enantiomer is kept away from the pharmaceutical. Thalidomide is an example of a sedative drug that was provided in the 1950s and 60s to pregnant women. It was thought to be safe. However, thalidomide prevented the proper growth of the fetus inside the body, and it resulted in horrible birth defects. The drug was marketed as a 50-50 mixture of two enantiomers. One of the molecules was enantiomers was a sedative. The other was found later to cause fetal abnormalities. Understand the problems of drug delivery and some simple solutions. Drug delivery means how a pharmaceutical is provided to a patient, whether it be a human or other mammal. Most oftentimes, they're delivered orally by inhalation, by breathing it in, by a patch put on the skin, by a suppository, something put in the digestive system, by drops, or by injecting the pharmaceutical directly into the bloodstream of the mammal. Now, in drug delivery, the physician, whether it's for an animal or for a human being, should know about the drug and the effects of the drug on the specific individual. They should know the personal history of the patient. They should also know about some of the abnormalities that might happen and tell the patient about that. They need to know the side effects. They need to know the right amount of the drug to administer and the best time to administer. They need to know if the patient has a tolerance for the drug, or if the patient has had a dependence upon or will gain a dependence upon this drug, or will have withdrawal symptoms. Of course, all of this drug delivery is based upon the correct diagnosis of what's wrong of the patient by the physician. The next video shows a new smart way of drug delivery that has recently been beta tested.
Pills, liquids, needles, patches. There are all kinds of ways to get medicine into our bodies. Now, scientists at Texas A&M University have discovered a new method that could change drug delivery. It's what biomedical engineer Mike McShane calls a smart material.
Smart materials are materials that respond to their environment.
The material is a gel that contains dye and microcapsules. Each of the tiny capsules can hold a sensor or drugs that can be released into the body. Inside the body, the gel becomes a solid, and an optical device allows researchers to see the dye in the material through the skin.
The light that's emitted back is measured, and that tells us what's happening in the tissue.
The material can hold and dispense various doses of different drugs and can even target certain areas of the body. It would allow doctors to control how fast and how much medicine a patient receives.
So we can actually produce cocktails of drugs that would work over a period of time, giving different amounts of drugs at different periods of time.
The material can also be used to monitor diseases like diabetes. It can pick up information like how blood sugar is being regulated in the body.
Basically, what we're talking about is collecting the kind of data that we've never had access to before.
A material that could change the way drugs are delivered and diseases are monitored for millions. I'm Josh Lebowitz, reporting.
Here are some questions to start your thinking. Again, these two questions, although not specifically chemistry equation related, are typical of the kinds of questions you would see in paper 2 of the Cambridge examinations.